And is it moving also?
No,
Yeah.
it's not moving.
It's moving now or no?
It's moving now.
Yes, it's
Yes.
moving.
Okay. So, yeah, this is about case 4 lecture, where we are going to know about the what is the basic genetic mechanism, or what happens at the molecular level in the cell, and then a normal cell turns into a malignant one. Okay, so this is called as a genetic basis of cancer or molecular basis of cancer. Or in a simple way, we know there is a normal cell growth. A cell divides from 1 to 2, 2 to 4, 4 to 8, so on, whenever there is a requirement. But in cancer, this cell growth is an abnormal one. So, the intending learning objectives for this lecture is a basic pathology of the tumor. Like, I'm not going to talk about much in detail about what is benign tumor, what is malignant tumor, what is like the nomenclature of tumor. When do you call carcinoma? When do you call sarcoma, adenoma, adenocarcinoma? This you have already taken. Okay, we just see what is the definition of neoplasm and what is the basic pathology of the neoplasm. Then we will touch a very basic concept, because to go in detail, the one lecture will not be enough to know about the cell proliferation and carcinogenesis. Okay, so we'll just see a basic outline of the cell proliferation and carcinogenesis, then knowledge about the sporadic and inherited cancer. When do you call it is a sporadic cancer, and when do you call this cancer is a inherited one? Okay, and what are the predisposing genetic factor or chromosomal abnormalities that will lead to a development of the cancer? It could be a mutation in the somatic cells, or it could be mutation in the germline mutation. There is roles of different genes. Remember, these 4 genes are very important: oncogenes, tumor suppressor genes, replication error gene, or the gene that will like lead to the limitless replication of the cells, and the gene that repairs the DNA, and a basic knowledge of the spread and the clinical effects of a tumor. Okay, this lecture you might not find something in detail, because the last year there was like a time constraint. We could not finish this lecture on time, so I have modified this lecture, and you will be given the same lecture with the additional slides as an additional resource. If someone wants to read something more in detail, they can go to that additional resource lecture, where you have got some more details about the genetic basis, carcinogenesis, and spread and the clinical effects of the tumors. So these are the basic ILOs, where we are going to know about some basic concept. Because repeatedly I am telling, we are just going to touch about the basic concepts. Because to go in detail, it requires a PhD in one part of a single gene. So it is out of our scope to know everything in detail over here. Something you must be knowing about the tumor genes in your life cycle case 7 lecture, when Dr. Abdul Fattah Al Ayubi gave you a lecture about the gene that works into tumor genes. Okay, most of the names of the gene will get repeat over here, and the functions I will not talk in detail. We'll just touch the basic concepts. So what is the basic pathology of a tumor? The tumor term was originally applied for the inflammatory swelling. You know the 5 signs of inflammation: tumor, dolor, calor, rubor, and the loss of function. Where the tumor means swelling, dolor means pain, rubor means redness, calor means heat, and the loss of function. These are the 5 signs of inflammation where tumor originally means the swelling. But nowadays, no one uses the term tumor for the swelling. So whenever you call or you heard a word tumor, it is always a neoplastic one. So what is a neoplasm? This definition, if you understand this definition, it will be very easy for you to understand the concept of a neoplasm. Okay. So what is a neoplasm? It is a mass of tissue, but that mass is abnormal. So this is an abnormal mass of tissue. Suppose you get a tumor in colon. This is nothing but the colonic tissue. A tumor is nothing but a colonic tissue mass, but this mass is abnormal. And the growth of this mass is very, very excessive and uncoordinated. What do you mean by excessive and uncoordinated growth? Suppose a cell take 24 hours to divide into two. Here it will take less than a minute or less than an hour. So this growth is very excessive as compared to the normal time required. What do you mean by uncoordinated? Uncoordinated means there is no coordination. One cell divide into two, two into four, four into 8, 16, and so on. Here, 1, 4, 8, 20, 100, like that. So the growth is excessive and uncoordinated. Okay. And this growth is persistent with the same excessive manner even after the cessation or stopping of the stimuli which cause this growth to happen. So for example, I give always this example. One, a smoker developed the lung cancer and he knows that this cancer has come from the smoking. So he asked, Doctor, can I stop my smoking and my tumor will stop? No. Once this process is has initiated, it cannot be stopped even if you remove the stimuli. Same happen if someone is working in the radiology department, he get exposed to the radiation and now he develop some kind of leukemia. And then he asked to the chief of the services, oh no, I have developed the leukemia. I need to stop my leukemia and I have to stop working in the radiology department. No, once the initiation has occurred, it cannot stop the growth. So, excessive, uncoordinated, persistent growth that will keep on growing in the same manner even you stop the stimuli. Okay. When do you call a tumor it is benign? When the microscopic and the gross characteristic are relatively similar to the tissue of origin. And clinically, the tumor remain localized. It will not disrupt the surrounding structure and it will not go from the organ to another places. So that time we call it is a benign or innocent tumor. While the malignant tumor are exactly opposite to the benign one. They grow excessively, they do not remain localized, they invade or infiltrate the surrounding tissue, destroy their function, and they have a capacity to go through the blood or lymphatics from one organ to another organ. And when you heard the terminology cancer, it always means a malignant neoplasm. So all these terminologies are clear for you?
Yes.
Okay. So when you heard it's a benign tumor, you take a breath of relief. You sigh for a relief. Okay, now this is a benign tumor. A simple surgical excision will cure. It will not go to the distant organ and it will not affect the life of a person. But when you heard a word of cancer or a malignant tumor, you know you have to be worried and you have to go accordingly and manage aggressively this malignant tumor. Now, what are the etiology? What causes tumor? Majority of the time, you will not know what is the exact etiology of a tumor. You see the, like, yeah, we keep always telling that lung cancer is very common in the smoking, alcohol causes the liver cancer, and so on. But you see a non-smoker who has never smoked, who has a very clean healthy lifestyle, may come up with a lung tumor, and he also doesn't have any history of the lung tumor in his family. So majority of the time, you don't know what is the exact cause for a tumor. But there are few documented causes for the tumor, like we see the inherited cancer. When there is inherited genes, or sometimes some viral infection or viral gene get incorporated in the susceptible host oncogene, some such as the MYC gene, which get infected by the Epstein-Barr virus in Burkitt lymphoma. MYC gene in the Burkitt lymphoma is a very good example for the inherited gene. Other thing you will know, the APC gene, adenomatous polyposis coli gene, neurofibromatosis, neurofibromatosis type 1 or type 2 gene. These are all the inherited type of the genes. Chemical carcinogenesis, exposure to the chemical, is a well known and very well documented uh, etiology of a tumor. This chemical can be a direct acting one or the indirect acting one. There are a group of the chemicals like polycyclic hydrocarbons, which causes sarcoma. From where they get, someone get exposed to the coal dust, gasoline, or the benzene, or the petroleum products, they have high chances of the development of the sarcoma. Aromatic amines, like they are used in the various industries like benzylamine, aniline, they causes carcinomas. While alkaline nitrosamine, they might be seen in the flavorful food. You eat the flavors to the food. Sometimes you get the juice, asir. You see it is a mango juice, okay? But they, most of the time, they don't use the original fruit. They use the flavors, okay? So these flavor foods can lead to the different causes of the cancers, and most commonly the hepatocellular carcinoma. So these are the direct acting. There are some indirect acting also, like the aflatoxin B. They get, like, sometime the fungus, especially the fungus aspergillus fungus. They infect the grains or the nuts, especially the fool Sudani. When you eat the infected fool Sudani, these aflatoxin get incorporated in the human body and they cause hepatocellular carcinoma. The another documented carcinogen is the physical, like you get exposed to the radiation, like the ultraviolet radiation. That's why they cause the skin carcinoma. That's why they always say, whenever you're going to the sunny environment, always apply a sunscreen so that your skin can get protected from the harmful ultraviolet rays and you can be protected from carcinomas or melanomas of the skin. Then radiation, like in the working in the radiology department, or someone get exposed to the nuclear radiations, okay? So they have special type of the tumors like leukemias, lung cancers, the one who works in the mines of the uranium or the radioactive minerals, okay? There is also exposure to the some environmental factor like asbestos. This causes mesothelioma. Another one is microbiological, like some viruses in the bacteria. Could you please tell me which bacteria can cause the cancer?
EBV,
Bacteria.
H. pylori.
Yeah,
Helicobacter.
Helicobacter pylori. Yeah, it's very well known to 80 to 90% association with the gastric carcinoma, 100% association with the lymphomas, especially the mucosa associated lymphoid tissues, lymphomas. Then we have these viruses. Like there is DNA viruses and the RNA viruses. DNA viruses, you have human papilloma, herpes zoster virus type 2, hepatitis B virus, Epstein-Barr viruses. You have RNA viruses like hepatitis C viruses, human T cell leukemia, lymphoma viruses. Okay, so these are the etiology of the tumor. Now we go for the, with this etiology, what fundamental changes or what changes occurring in a cell that lead to the development of a cancer. So basic fundamental or basic principle or core principle of carcinogenesis is the non-lethal genetic damage. What do you mean by non-lethal? See, normally whenever there is a damage, genetic damage to the cell try to repair itself. And when it is not able to repair, the cell will go into the programmed cell death, that is known as the apoptosis. So that the genetically damaged cell should not come into the cell division. Only a normal healthy cell should come into the division. But what happened in the carcinogenesis? These genetic damages are so severe that they make the cell to lose its ability to go into the repair or the apoptosis. So they become non-lethal. They become like immortal. They cannot die. So these non-lethal genetic damage is the basis of the carcinogenesis. And these genetic damages or mutation may be acquired by environmental agents like the chemicals, physicals, or microbiological, or they may come through the inherited one. Okay, there are very important there are 7 classes of actually gene, or 7 or 8 classes of the gene, but these 4 classes of the gene are the principal target of the genetic damage. What is these gene? The growth promoting proto-oncogenes, the growth inhibiting tumor suppressor genes, genes that regulate apoptosis, and the gene that involved in the DNA repair. So these are the 4 targets of the genes which get affected in the tumors. And remember, tumors are monoclonal. What do you mean by monoclonal? It means there is damage to one single gene. One single cell has get affected. And these affected single cell will proliferate and form a mass of tissue. So this is always arise from a single cell. So tumors are monoclonal in origin. So before going into more detail, we see the normal cell proliferation. How a cell proliferate normally. Whenever there is a need of cell to undergo division, there is a growth factor. These growth factors, suppose a platelet wants to undergo in the division, there will be a platelet derived growth factor, or a fibrous tissue needs to undergo division, there is the fibroblast growth factor. Okay, so these are the some growth factors, specific growth factor, they get activated and they bind to their specific receptor. These receptor then get activated for a limited period of the time till there is a need of the growth. Once the growth is sufficient, your need is uh, fulfilled, the activation of these growth factor receptor will stop. So this is transient and limited activation of the growth factor receptor. Once that get activated, this will activate several signal transition protein in the inner side of the cell membrane. So once from outside of the cell, it binds to the receptor get activated, it lead to the inner side of the cell membrane, from the cell membrane through the cytoplasm with the different mechanism like the second messenger or the activation of the second different pathways, it goes to the nucleus. And once it reaches the nucleus, it goes to the DNA and tells the DNA to undergo division. This is all how the normal cell divides. But in the abnormal tumor cell, everything's become abnormal. The growth factor is abnormal. There is like for a longer period form and the infinite time of the activation of the receptor, there is signal transduction that is also abnormal. There is genes that lead to the second messenger that get activated or that get mutated, and there is also mutation in the DNA proliferation, so that is abnormal. So when you go to the molecular basis of the cancer, you must know cancer is a genetic disease because there is the principle of the cancer is the non-lethal genetic damage. So cancer is a genetic disease. This genetic injury or injury to the genes or the DNA may be through the environment or through the inherited one. And tumors develop in a monoclonal expansion. Okay, there is a clonal expansion of the single genetically damaged cells, and the four classes of the gene. The principal target of these genetic damage is mainly on these four classes: oncogenes, tumor suppressor genes, DNA repair genes, and the genes that regulate the apoptosis. Once these genes get affected, there is transformation of the cells from normal cell to the clonally affected tumor cells, and these tumor cell growth go into the progression from growth to the invasion to the metastasis. So tumor formation is not a single step, it is a multi-step process. So what is the hallmark of the tumor cells or the malignant cells or the tumor cell hallmark? Okay, so what happen when a tumor cell when a normal cell change to the tumor cells? So they become self-sufficient in the growth signals. They do not depend on any other factor. They are self-sufficient. As you say, like once you start earning, you no more dependent on your parents for the money. You say, now, Baba, I am self-sufficient, so I do not require your help. I can manage myself. Okay. But at the same time, they become insensitive to the growth inhibitory signals. Okay. So if a son is not obedient, he's not listening to his father well, his father asks you not to spend money over here, stop here. But he said, no, 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 I am self-sufficient. I do not need any inhibition from you. So they become insensitive to the growth inhibitory signals. So with these two things, self-sufficient in growth signal, insensitive to the growth inhibit, they become, they start dividing. They go into the progression. They go into the multiplication. Still there is something like, suppose say mama, he listens something to mama. So mama will ask, okay, don't do this. This is the role of DNA repair. If he listens to them, this all the abnormal cell will get repair and they become the normal cell, and normal cell come into the mutation. But after a time, they become resistant to this also. So there is dysfunction of the DNA repair genes. Still, there is a chance of something like the apoptosis, the programmed cell death. That this is the abnormal cells. There is abnormal oncogene, abnormal tumor suppressor gene, abnormal DNA gene. So there is some role of apoptosis. They try to stop this abnormal cell to go into the division. But this cell is very important, very, very, like what we call is a shatir. Okay. So he evade this apoptosis also. And it acquires one more thing. It goes into the limitless replicative capacity. Suppose a cell can divide only up to 100 times, but it keep on dividing infinite time, maybe million or billions of the time. Now what happened? This is growing, 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 growing. It needs more blood supply, more nutrition. So now they start developing their own blood vessels. So they get their sustained angiogenesis. If these angiogenesis is not there, they will not get the nutrition and they might die. But they are very important. They are very, very shatir, as I told you. They are very intelligent. They get their own blood supply. Now they are getting their own nutrition. They keep on growing. Now they go into the surrounding tissues or they go into the distant organ. And they also have the escape from the immunity. Now the normal immunity mechanism also cannot help them. So, do you understand this hallmark of the tumor cells?
Yes, doctor. Yes.
Okay. So, cancer cells behave in this abnormal manner because there is a mutation. And these mutation mainly in the level of the DNA sequences. And these are the genes which are affected, like oncogene, tumor suppressor gene, DNA repair genes, genes for the apoptosis, genes for the replicative capacity. There is genes for the blood vessel formation. There is also some genes which lead to the invasion and the metastasis, and some genes which also escape from the immunity. So there is different mechanism, like they get the self-sufficient in the growth signal, they become insensitive to the growth signals or insensitive to the tumor suppressor gene. Okay, there is escaping from the apoptosis, there is limitless replication, there is tissue invasion and metastasis, there is sustained angiogenesis, there is defective DNA repair, and all these put together, a single cell goes into formation of a tumor. Now we go something about, now we are talking about the oncogene, the growth inhibitory genes or tumor suppressor genes, genes that regulate DNA repair or apoptosis. What are those? Okay. So we go to the first we go to the oncogenes and the tumor suppressor genes. Okay. These are the oncogenes are the normal genes. When they are normal, they known as the proto-oncogenes. And these proto-oncogenes are essential for the cell division, normal cell division. If these genes are not there, cell will not divide. Okay, but they divide at a specific level. Once the division is over, there is a role of tumor suppressor genes. These are the normal genes which inhibit the cell division. Okay, you required a cell to divide only up to 100 cell level. Once this is achieved, this tumor suppressor gene comes, they send the inhibitory signals, and this process stop. Okay, so and they usually work in this phase, G1 to G0 phase. Okay, so you have the stimulating factors like growth factor receptors, and you have the inhibitory factors or the inhibitory genes such as P53 or RB genes. Okay, these are the genes which keep a check from the G1 to G0 phase, and they remain in a balance so that there should not be any abnormalities. Okay, so this is the normal. There is a normal number of the inhibitors, normal number of the stimulators, and this is the normal growth which happen in cases of the repairs, which happen in cases of the organ, like when the fetus is forming in the uterus, or whenever there is an injury, it happens over there. Okay, but in the cancer cell, this balance is lost. There is less inhibitor and more stimulators, or there is a less tumor suppressor gene, or less inhibition from the tumor suppressor gene, and the more and more stimulation from the oncogenes. So cells keep on proliferating, and this is an abnormal genetically damaged cells which goes into the cell cycle, and there is a growth and the formation of a tumor mass which invade and metastasis. So this is like how normal cell, whenever there is a DNA damage, either there is a successful DNA repair or apoptosis. When the DNA repair is successful, a normal cell will go into the division. When the DNA repair is unsuccessful, it either go into the apoptosis or it remain a DNA damaged cells. Okay, so whenever there is a DNA damage, there is failure of DNA repair. Okay, and this comes from the inherited mutation in the gene that regulate the DNA repair or apoptosis. So there is mutation in the genome of the somatic cells, activation of the growth promoting oncogene, inactivation of the tumor suppressor gene, or alteration in the gene that regulate the apoptosis. So when there is these two mechanism, unregulated cell proliferation. When there is this mechanism, alteration in the apoptosis, there is decreased apoptosis, no programmed cell death. This all lead to the clonal expansion of the cells, and once this expansion happen, there is angiogenesis, there is escape of immunity. Because this is an abnormal cells, whenever there is an abnormal cell, there is high chances of additional mutation. These additional mutation can come from here or can come from some other things. So there is progression of the tumor growth, malignant neoplasm, invasion, and metastasis. So this is how there is like division of an abnormal cell that lead to the development of a tumor which invade and metastasize. So what are the oncogenes? Okay, these oncogenes will lead to the self-sufficiency in the growth signal. They become like autosufficiency. They don't need any dependence. Okay. So what are the example? You have growth factors such as platelet-derived growth factor. And what is the tumor? Glioblastoma. Growth factor receptor, you have RET. This is usually seen in the multiple endocrine neoplasm type 2A and type 2B, and familial medullary thyroid carcinoma. Receptor tyrosine kinase c-KIT. This is also a growth factor receptor abnormal gene. When it get abnormalated, you get GIST, gastrointestinal stromal tumor. ARB-B1, squamous cell carcinoma of the lung, glioblastoma, head and neck. ARB-B2, the another name for ARB-B2 is HER2-neu, and you know this is seen in the breast cancer and some adenocarcinoma of the ovary, lung, stomach, and the salivary gland. So here you need to know the example and what tumor cause. For suppose growth factor, you need what is the gene responsible for the growth factor. You need for the PDGF, EGF, platelet fibroblasts, epidermoblast, growth factor genes. Receptor, you have RET, c-KIT, RB1, RB2, and the important cancer caused by abnormalities on these genes. Signal transduction protein, you have KRAS, HRAS, and RAS. KRAS in the colon and the pancreatic carcinomas, HRAS in the bladder tumor, and RAS in the hematopoietic tumor like leukemia, lymphoma. Nuclear regulatory protein is the MYC. You have there is a dysregulation of this regulatory protein. There is NMYC, LMYC. So you see in the neuroblastoma and the small cell carcinoma, and the CMYC in the Burkitt lymphoma. Cell cycle regulator like cyclins and cyclin dependent kinase. You have cyclin D genes, which if it get mutated, you get the breast cancer, esophagus, liver, and the lymphoma. If you have the germline mutation in the P16, you get melanoma. If you have somatically acquired mutation in the P16, you get pancreatic carcinomas. So why I'm not going in detail. These are the simple thing but important. There is name of the genes and the commonly affected tumor seen when these genes get mutated. So you remember the name and remember the common tumors which seen when these genes get mutated. I know this is the long, but you have to remember. You might get questions over here in surgery and definitely in the SMLE. So this is how the growth factor bind to the receptor, signal transduction, goes through the cell, come to the nucleus DNA repair, or the genes that uh, promotes apoptosis. There is some transcription factor, cell dependent kinase or cyclines, cyclin D1, cyclin D2, cell regulators. So these are all just like a pictorial presentation of these what we discussed so far. So growth factor receptor, signal transducer, transcription factor, and cell cycle regulator. So these are the 5 example. One growth factor like you have PDGF, TGF beta, you have growth factor like EGF receptor, RET, or something like that. You have signal transducer, RAS, you have cell cycle regulator, cyclin D, cyclin DK4, you have transcription factor like MYC, CMYC, LMYC, NMYC, and you have, so these are the 5 classes of the genes, oncogenes and their example and their cancers. Tumor suppressor gene, these are the genes that are normally genes which inhibit the cell cycle or they switch off the cell proliferation. Once the required number is achieved, they go and ask to stop and they generally act in the G1. This is a tumor suppressor gene. So we need loss of both copies. You know, any gene has 2 copies. So here loss of both copies are required to develop a cancer, while oncogenes are the dominant type 1. So even a single copy of a gene is affected, the person have chances of development of the carcinoma. So tumor suppressor gene require loss of both copies, while oncogene require loss of only one copy. And in the tumor suppressor gene, what happened? Only one defective gene is inherited or one defective copy is inherited. And malignancy develop when other allele losses function with a somatic mutation. This is known as two-hit hypothesis. Suppose a child is born, one copy of the RB gene affected, and he get exposed to some environmental oncogenes or environmental carcinogenetic factor like ultraviolet radiation or something like that, the second copy get affected. This is called second hit. And now he is susceptible to develop a full-blown malignancies. While in the oncogenes, only loss of function, one copy, and there is a loss of function which can lose to the sensitivity to the inhibitor genes. Okay. So here they work in the G1, they switch off the cell proliferation, they need loss of both copies for the tumor suppressor gene. While oncogene, they need only one copy to get affected. And there is a two-hit hypothesis in the tumor suppressor gene. One is inherited and one is acquired. And in contrast to oncogene, tumor suppressor genes are the loss of function genes, while oncogenes are the gain of function. So here loss of function, it is simple because their function is to stop. So there has to be the loss of function. So this function to stop is loss. So there is no one to stop. Like Shurta. Shurta is there to stop. If there is no Shurta, you run and you go in excessive number. Okay. So like that. So there is a loss of function for the tumor suppressor gene. The prototype example of the tumor suppressor gene is the retinoblastoma gene. while the example for the familial one are the NF1 as seen in the neurofibromatosis, APC seen in the adenomatous polyposis coli, and sporadic cancer may result from the somatic loss of both allele of these genes. Okay, so what happened in the inherited? There is loss of one copy, and the second loss occur in the two hit mechanism. While in the somatic sporadic cancer, there is loss of both the copies due to mutation in the somatic cells. So these are the tumor suppressor gene P53, location of the chromosome 17, and the some important tumors. RB located on the 13, retinoblastoma. Neurofibromatous type 1 located on the 17, neurofibroma, malignant peripheral nerve sheath. APC chromosome 5, colonic carcinoma. WT1 chromosome 11, Wills tumor and the bladder carcinoma. Now we go to the P53 gene. While RB gene is the prototype of the tumor suppressor gene, and P53 known as the guardian of the genome. It is the main policeman or the main guardian of all the genome. If it is working properly, it will send the inhibitory signals and the cell cycle will undergo arrest and there is no abnormal cell proliferation. But when this gene gets affected, there is lot of cancer can happen. So see, abnormality of the P53 genes are found in the 30 to 50% of the all cancers. This P53 gene can affected into the germline or inherited one, and this is known as the Lee-Frommani syndrome. And this Lee-Frommani syndrome is the due to the germline mutation in the P53, and there is a like it is a syndrome, so you'll find many malignancies in this. Okay, so whenever there is a radiation expression of P53, there is a minor damage. P53 stops cell cycle till the DNA is repaired. If there is severe damage, P53 brings the apoptotic gene and it leads to the program cell death. But whenever there is a damage or like very excessive amount of radiation, like in the nuclear exposure, so when there is the P53 gene is unfunctional, there is a loss of function, so there is like no DNA repair, no apoptosis, so these damaged cells will keep on multiplying and forming of a tumor. So this is a better whenever there is a damage through any etiological factor, when the normal P53 is acting, this will get activated. It causes cell cycle arrest, bring in the DNA repair gene and the successful DNA repair and the normal cell undergo division. If the DNA repair, if P53 is not activated, no cycle arrest, no DNA repair, still it can bring in the apoptosis gene and it can lead to the apoptotic gene. But when there is continuous loss of P53 seal. Okay, there is a DNA damage. P53 is not activated, no cell cycle arrest, no DNA repair, clonal expansion of the mutant cells, and there is formation of a tumor. So see, the P53 is very, very important. Even if it is not get activated, but it is normal, it will bring the apoptotic gene and can lead cell to undergo a programmed death. But when it is mutated, there is no chance of these activities. Only thing is that a mutated cell will undergo expansion and form a tumor. So you are clear with the functions of P53 gene?
Yes.
Yeah, and that is why it is known as the guardian of all the genome. Now we have the gene that regulate apoptosis. These are the gene which is BCL2 or BEX2 gene. When they get overexpressed, they will lead to the special type of a tumor, which is known as the follicular lymphoma. Then gene, if they get affected, that regulate the DNA, so there is no DNA repair, and an abnormal cell will go into the proliferation. Okay, so it is also need to the both the copies of the DNA repair should get lost. Okay, and there are three types of these DNA repair gene. Mismatch repair gene, which is seen in the hereditary non-polyposis cancer syndrome, HNPCC. Nucleotide excision repair, you see in the xeroderma pigmentosa. Recombination repair, you see in the ataxia telangiectasia and the Bloom syndrome. BRCA1 and BRCA2, located on chromosome 17 and chromosome 13. They are actually a DNA repair gene, but they act as a tumor suppressor gene, and they are affected in 80% of the familial breast cancer, and they are not associated with the sporadic breast cancer. Okay, so BRCA1 and BRCA2 specific for the familial breast cancer. So this is the DNA repair gene, their example, and their associated tumors. Limitless replicative potential. You know, a normal cell has telomeres, okay, at the end. And with each division, these telomeres get shortened, and at the end, they get totally lost, and stop codon comes, and the cell stop dividing. Okay, but these telomeres are the enzyme. They keep on adding. The tumor cell acquire this enzyme telomerase, and they keep on adding a short portion of DNA to the affected cell. So with each division, there is no loss of DNA. Instead, there is addition of the DNA, and there is no stop codon, and there is no stopping of the cell division. So cell keep on dividing for the infinite time. Okay, so this is also one characteristic of the tumor cell, to get the limitless replicative potential with the help of telomerases. Then tumor angiogenesis, as I already explained, these tumor cell require nutrition, and this nutrition comes from blood vessel. But a normal blood vessel has a limited blood supply. So these tumor, to achieve their nutrition, they develop their own blood vessels. And there are the genes. These are the genes like VEGF genes, or angiostatin, endostatin, and vasculostatin. These genes comes into play. They get their own blood vessels, and these blood vessels are the abnormal blood vessels. So cancer is a multi-step process. There is not a single gene. There are the multiple genes at the multiple level. There are oncogenes, there are tumor suppressor genes, there are DNA repair, apoptosis, angiogenetics. Even once they have formed a full-blown tumor, there is additional mutation which comes that lead to the infiltration. There is one another mutation comes that will lead to the metastasis. Okay, so these are the, and the adenoma carcinoma sequence is a characteristic example for the multi-step carcinogenesis. There is normal epithelium which goes hyperplasia, adenoma, dysplasia, and the carcinoma. This we are going to discuss in detail in the next lecture. Now we go to the another part of the our ILO. What do you mean by the sporadic and the hereditary cancer? Sporadic cancer are seen in general population where they don't have any history of the cancer in the family. Similar cancer is in the family, and they do happen either somatic mutation, either due to the spontaneous or the induced. While hereditary cancer, they are less in number, 5 to 10%, and they are seen similar cancer in the families, and that happen mutation in the germline or the germ cells. So what are the characteristics of the hereditary cancers? These cancer often occur earlier than the sporadic cancer of the same cancer. Suppose take the colorectal carcinoma. If someone has the tendency of the hereditary colorectal carcinoma, they will develop a decade earlier, maybe say like 30 or 40 years of the age. While someone they don't have the hereditary predisposition for the colorectal carcinoma, there is like spontaneous somatic mutation, that is sporadic cancer, they are seen in the later ages like 60 or 70 years. Okay, so for the hereditary cancer, screening from the younger age is recommended. So for a normal population, you ask screening after 50 years. In cases of the familial cancer, you start from 10 years or 20 years of the age. And the hereditary cancers have the more than one type of the cancer. And they also affect the cancer treatment option. They need the aggressive treatment, and there is aggressive follow-up care, and the survival rate is also less in case of the hereditary cancer than the sporadic cancer. And hereditary cancer usually in the site specific, like colorectal carcinoma, breast carcinoma in the family, prostatic carcinoma, and you see generally two or more relative affected in this. So, but remember, cancer is a very common disease. So, some families will have some members who having the same cancer. Suppose like breast cancer, some says like auntie is having breast cancer, wife is having breast cancer, something like that. So, but it doesn't mean that is a familiar or hereditary cancer. This is the sporadic cancer. So, this is the hereditary cancer syndromes, affected gene, and the what type of a tumor you see. Okay, Lee-Fromeni syndrome, it is due to the T53 or tumor suppressor gene, P53 gene. The cancer, breast carcinoma, soft tissue carcinoma, adrenal, brain tumor, leukemia. Cowden syndrome or Bunyan-Zonana syndrome, it is due to the mutation in the PTEN. You have breast carcinoma, glioma, and the hematoma. Hereditary breast and the ovarian carcinoma, BRCA1 and BRCA2. So you have male breast carcinoma, male prostatic carcinoma, ovarian carcinoma. Definitely for ovarian carcinoma will be there. It's not written over here. Pancreas, Fanconi's anemia, and the brain tumor. Lynch syndrome, it is due to the affection of the DNA mismatch repair gene, MLH1, MSH2, MSH3, MSH6. You have colon, stomach, small bladder, multiple endocrine neoplasma type 1. The affected gene is menin. You get CA thyroid, CA adenoma of pituitary, carcinoma or adenoma of the pituitary, carcinoma pancreas, MEN2 due to the RET. You have thyroid, parathyroid, and adrenal gland pheochromocytoma, Peutz-Jeghers syndrome, SK2. So these are the important hereditary cancer syndrome, their affected gene and their tumor phenotype. Okay, you might get a scenario for that. You might have the type of tumors. They might say you the gene affected, ask for the syndrome. They might have the gene. They may have directly the syndrome. They might ask you the what gene is affected. They might have this syndrome. They might ask you what are the tumor most commonly seen over here. Okay. So here we finish our lecture of the abnormal cell proliferation. You need anything extra, if you don't understand anything, you can call me, you can ask me on WhatsApp. Inshallah, I will try to clear your doubt. Now we take a break for 5 minutes.